Okay, so today I'm talking about DNA replication. And so um, Synthesis of polynucleotides, as we've mentioned before, always involves adding a new nucleotide here or adding a C which matches the G in the template. So this is the template strand, this is the newly synthesized strand. Here's the next nucleotide. It comes in as a triphosphate nucleotide, so there's three phosphates. And the chemical reaction is making a covalent bond between this phosphate and this oxygen. And this oxygen is the hydroxyl group at the 3' position of the previous nucleotide, which was an A. So here's the deoxyribose sugar, and this is carbon 1, 2, 3. So the 3' hydroxyl is going to accept the new one. And you can only do this reaction by adding it to that 3' side. It forms a phosphodiester bond, which is shown here for the previous one. So the previous nucleotide was an A. It formed this covalent bond right here. The energy to make that bond, and so the energy to make a new polynucleotide, is from the high energy phosphate bonds. So these bonds with these three phosphates together are very high energy. So when you break this one to form that covalent bond, it releases a lot of energy, and that's the energy used to synthesize DNA. And then subsequently, the leftover two phosphates, which is called a pyrophosphate, that gets cleaved, and so that releases more energy. And so that makes the reaction irreversible. So once you add it, it stays on there. It doesn't tend to go back. So to make a new DNA molecule, you separate the old DNA molecule and you use each of the old strands shown here in yellow to make a copy of a new strand. So here we're synthesizing the new strand. The new nucleotide is always added to the 3' end, so we're going from 5' to 3'. The enzyme that does that is DNA polymerase, and that polymerase can only do this reaction. It can add a nucleotide. It cannot add a nucleotide to the 5' end. It needs this 3' hydroxyl. The newly synthesized strand, the red strand here, is always anti-parallel to the template strand, which is the yellow strand here. So you see the template strand goes 5' to 3'. The newly synthesized strand has to go the opposite. So it has its 3' on this end, 5' on this end, as compared to the 5' and 3'. So you can see these are opposite. So these are going, this is 5 to 3, and this is 5 to 3 in that direction. And so whenever polynucleotides bind together, they have to be anti-parallel. The way that DNA is synthesized is by separating the strands and making a copy of the old strand. So you end up with two new DNA molecules, each of which has an old strand and a new strand. And so that synthesis process is called semi-conservative, where you split them across, you split it open, and then make two new strands. So each new DNA molecule contains one old and one new strand. You can see that here. So here's your original DNA molecule, you separate it, and then you make copies. And so you end up with two DNA molecules. You replicated the DNA into two copies, and each which has an old and a new. So remember what we're talking about in this chapter. So the previous chapter, we were talking about transcription, right? And that's making a messenger RNA copy of a single gene. So it's a very short, relatively short job, right? It's just copying one little segment of a chromosome. DNA replication is making complete copies of all the chromosomes. So in humans, that's 46 copies. It's billions of base pairs. So this is a huge job. You have to make. So if this was a chromosome, it would extend way to like Baton Rouge and to Slidell in both directions. So DNA replication is a big job, and the product is two DNA molecules, where transcription is a small job, and the product is a messenger RNA. And so because this is such a big job, in organisms like us and other eukaryotes, it starts at many places along the chromosome. So DNA is a very long, stable molecule, but the individual bonds that hold the two strands together, which are the hydrogen bonds between bases, between A's and T's and C's and G's. So you can pull apart little parts. You're just breaking a few hydrogen bonds. So it's not hard to pull apart the DNA molecule at a little segment. It's really hard to pull it completely apart when it's millions of base pairs long. So the way we start replicating is pull apart a little section and start replicating there. Um, so that's what all says. And so these spots are called replication origins. And so even though it would be hard to split the two strands together, just doing a little bit is easy. And so if we're going to copy all of the chromosomes, it's better to start at multiple spots and then proceed from there. So here we have two origins of replication. We've pulled the two strands apart. And then we're going to start copying in both directions. So we pull it apart, copying this direction and copying that direction. And you can see, here's a third one that opened up. So we're copying the new strand as the red strand. And we've started at this beginning, and now we're going out. So here we started here. So we started right here, and then we started copying, copying, copying. And we went in both directions. So we go in both directions for both strands until eventually we keep copying, and then those origins meet. And at that point, we'll have copied the whole chromosome. So the way that DNA replication appears is you start at these origins of replication and then synthesize both strands in both directions from that origin. So each side of these origins of replication are called a replication fork. So this is a fork and this is a fork. So if we zoom in on this particular fork, so again, the overall direction that we want to copy is in this direction. So here's that fork. Here's our top old strand, our bottom old strand. Here's our newly synthesized strand. And we're going to copy in this direction. So we're copying both in that direction. So there's a problem down here. So if we want to copy both of these templates in this direction, what's the problem? Think about that for a second. Okay, so the problem is here. We can't add new nucleotides to the 5' end. So how do we copy in this direction? Right, we can only add nucleotides to the 3' end. Can't synthesize to the 5. You can't synthesize 3 to 5. You can only synthesize 5 to 3. You can only add new nucleotides to the 3' end. And so this is a problem. How do we do that? And so the way it's done is that each strand works differently. And so we have basically 4 different enzymes that are working here. One that's doing this top strand, one that's doing this bottom strand, and those are trying to go in this direction. And then another one that's doing this top strand going in that direction and this top strand going in that direction. And so there's kind of 2 mechanisms at work. This bottom one here, where the newly synthesized strand has its 3' end going in the correct direction so that we can continue to proceed, is called the leading strand. And that one, there's no problem. You just continually synthesize, keep going and going. The problem strand is called the lagging strand. And because we can't synthesize just continuously going in this direction, what happens is that the enzyme jumps out and then copies back towards where it came from. And then once that's done, it jumps out further, copies back, jumps out further, and copies back. The way that actually works is the template is pulled out into a loop and it copies back around the loop. But overall, it's synthesized in these little short strands that are all synthesized 5' to 3', which is opposite the overall direction you want to go. But then those are eventually filled back in. So for each fork, we have 2 enzymes working. We have the leading strand and the lagging strand on this side, and we have the leading strand and the lagging strand on that side. And so the solution, again, is synthesizing opposite the overall direction, but doing it in little short strands. Those short strands are called Okazaki fragments. And then an enzyme comes back around and links those together. That enzyme is called a ligase. So the lagging strand solves this problem with DNA replication by synthesizing things in the opposite direction, just little short strands, and then linking them back together. So there's one other issue that has to be overcome in that the DNA polymerase that does this reaction, adding a nucleotide to the 3' end, it actually needs double-stranded DNA. So it needs 2 strands. All it can do is bind to something that's already bound to a template and add it on. It can't work on just 1 strand by itself. And so the solution is that short little RNA primers are added, and that allows the enzyme to work. So RNA is allowed to be made with just a single strand because of an enzyme. And so if we add those primers, then the RNA polymerase can use those. For the leading strand, we only need to synthesize a primer once. For the lagging strand, you need to do it for each lagging strand, or each Okazaki fragment. And so this is a better visualization of that. So here is our last lagging strand. And so in order to make our next lagging strand, first a little blue RNA primer is made, and then DNA polymerase can make its Okazaki fragment. Then later on, the RNA is chewed up and then linked together with the ligase. And so all these blue ones are the RNA primers, and then the red is the DNA. So in order to complete the lagging strand, you have to both jump out, make the primer, make the lagging strand Okazaki fragment, and then link those back together using a ligase. So we've talked about several things about leading and lagging strands, and here is a review of the enzymes involved. And I'm also going to put up a video that shows an animation of this, which actually makes it a little more realistic for how things are working. So the enzymes that do DNA replication form big complexes at the replication fork. So each of these are the polymerase here and the little green things. And so the first thing that happens is there's a helicase shown here in yellow that separates the 2 strands. Then there's the primase, which is shown in this little blue. It's making a little RNA primer so that it can be added on to the lagging strand. So this is RNA primer. Now the lagging strand is being made. There's a sliding clamp, the little red shown here, and that clamps the polymerase down to the DNA. And these little brown arrows are single-stranded binding proteins, because when you pull out the lagging strand, you need to block it from binding back on itself. So the single-stranded binding proteins protect the lagging strand. And then the ligase links those fragments together. So watch the video, and I think it makes it a little clearer what's going on with those enzymes. The enzyme itself, DNA polymerase, is really accurate. And that makes sense because you're copying the whole genome, so you don't want to make a lot of mistakes, because that genome is going to be passed on to the next cell, and in sperm and egg, it'll be passed on to the next generation. So the DNA polymerase only makes an error 1 in every 10 million nucleotides added. And one of the reasons it's so accurate is it has its own proofreading activity. So proofreading is actually at a second catalytic site, and I'll put up another video showing a little cartoon of this. But so that the DNA polymerase has 2 enzymatic activities. One adds the nucleotides to the growing strand, and the other one can remove, to chop out ones that have been added incorrectly. So it proofreads itself. And so it's a very accurate enzyme. There's one last issue that comes about when you reach the ends of chromosomes. So here we're reaching the end of the chromosome, and now we encounter another problem. So here's the leading strand. We've copied all the way to the end. But here in the lagging strand, here's our primer. Here's our last Okazaki fragment. Once we connect all of these together, we want to fill in this last little gap, but there's nowhere to make a primer for that end. And so that would leave this little end of the chromosome uncopied. So polymerase can't make this little gap, and at the end, the chromosome would get shorter every time it was replicated. And so there's lots of cell divisions, and you replicate your DNA with every cell division. So your chromosomes will get shorter and shorter and shorter. So that's a big problem. And so that's solved by adding a bunch of repeats to the ends of chromosomes. So these little boxes are repeated sequences And the end of a chromosome that has all these repeat is called a telomere. So that serves as a protective cap, such that even if you don't copy, if you lose a couple repeats at the end, it doesn't matter because the information is still protected. And so the end of the chromosome is called a telomere that has those repeats. The telomere also allows the cell to determine if the chromosome is still intact, because if it breaks in the middle, then those middle ends don't have telomere sequences, so the cell knows that that's a broken chromosome. So the telomere also marks a chromosome as this is the end, this is what it's supposed to look like. So the enzyme that does that is called telomerase. It has a built-in RNA primer, and so here it's got double-stranded, and now it's going to extend the DNA strand in this direction. And this is the repeat sequence, so it's going to make that one go down, make the next one. And so it's just going to keep cruising down. It has its own little primer. So the telomerase enzyme just adds a bunch of copies to the end of the strand. And now you can come back, add your RNA primer, and copy all of those. And the only thing that you lose will be some of those repeats, which can be added on in the next round through another telomerase. Here's another cartoon showing that. So there's the telomeres being added by telomerase. Then your normal RNA polymerase and your ligase can finish copying the rest. Okay, so that's all of the parts about DNA replication. The last thing I wanted to introduce was a technique called the polymerase chain reaction, so our PCR. And so it's a technique that's used to make many copies of a particular segment of DNA. And it works by having primers that flank whatever sequence you want to copy. So in this case, these little blue ones are the primers. One is the reverse primer that has its 3' end going in this direction. The other is the forward primer that has its 3' end going in this direction. So here's our target DNA molecule. We split the strands, add our primers, and then this gets copied in this way, and this gets copied in that way, and we end up with two new molecules that have copied that segment. And so all PCR does is make many, many copies. So that's one cycle. Now you continue doing that over and over again, and you make billions of copies of that little segment. And so the 5' and 3' determines which is the reverse and which is the forward. And so in the end, the reaction is only going to copy the sequence between those primers. So you're going to end up with DNA molecules that are just between those primers, as you can see here. So it's just that segment, and you're going to get billions of copies of that little segment. The method involves cycling between temperatures. So this is one cycle, and here's the next cycle. And so why does the temperature help to do this reaction? So let's talk about it. So there's a cycle between 3 temperatures. So the first temperature is a high temperature, 95 degrees Celsius, and that serves to break apart the DNA molecule so that the two strands are separated. So this is almost boiling. So in order to separate two strands, especially for longer DNA molecules, you have to really heat it up to break apart those hydrogen bonds. The next thing you do is to cool the temperature back down, and what that allows is for your primers to bind. So if 95 degrees is separating the DNA molecule, you have to cool it down in order to get the little primers to be able to bind. They can't bind at that high temperature. So 65 degrees is called the annealing temperature because the binding of primer to the template is called annealing. And then you raise the temperature a little more, and that helps the polymerase work that's going to do those copies. So it's a specific kind of polymerase that can survive all these temperatures, and it works a little better at 72. So you go high to separate the strands, low to allow the primers to bind or to anneal, and then medium to allow the polymerase to work. So that's one cycle. You've separated the strand, added your primers, and then copied. Now, at the end of the cycle, first cycle, you have 2 DNA molecules. And now each of those 2 DNA molecules can now be templates. So after cycle 2, you do 95 degrees, annealing, copying. So at the end of that, you end up with 4 DNA molecules. So for each cycle, you double the DNA molecules. After 3, now you have 8 DNA molecules, and you continue to exponentially increase the number of DNA molecules. And you can see that as of the 3rd cycle, you get mostly DNA molecules that only are between the 2 primers. And that's because, once here, you can only copy that part. So you end up with basically all DNA molecules are just this little short segment between the 2 primers. And that segment becomes the majority segment. So I'm going to post another video which shows that a little better on how you get just this segment copied. But the overall goal of PCR is all you're doing is copying a segment of DNA between the 2 primers that you design. Now, PCR is used in many different applications, all kinds of different things. It's used for COVID-19 testing, anything that is a DNA sequence, PCR can be used to try to identify things. One of the things that's used in crime scene investigation use what are called microsatellite loci. And that's sites within the genome that we've found through sequencing of the genome, where there's a short number of repeats. So these are old mistakes that happened way back a long time ago and then has been inherited over time. And individuals can have different numbers of repeats at these sites. So here's individual 1. Here are the repeats. And so it has 8 of those repeats. The repeats, I think, are CAC. So CAC 8 times. That's individual 1. Individual 2 only has 2 of those repeats. Oh, no, the repeats is CACA. So there's 2 repeats, and here's another individual that has 8 repeats. And so this is, Johnny, this is Freddie, and this is Jimmy. And you can tell the difference between those by looking at how many repeats they have in their DNA. So it's a way to identify individuals. Now, you can't tell with just one, right, because both Johnny and Jimmy have 8. But if you do this for multiple loci throughout the genome, then it becomes very specific for that one person. So the microsatellites are also called tandem repeats. And we can analyze the number of how many this individual has at that particular spot by using PCR, because you design primers for just outside of the repeats, and then you make this little segment, and then by running on a gel, you can determine the size of the little segment that's made by PCR. So if you look at that data, this individual, when we do PCR with these 2 primers, we get a product that's 8 repeats long, which runs here on the gel. Here we get a product that's only 2 repeats long, which runs much further on the gel. And then here, we get a product that runs at 8. And so the size of the PCR product tells you how many repeats. And if you do this for many loci, like here, you get this collection of all different microsatellite loci sizes. And then at some point, it becomes very specific for one individual alone. And there's going to be a problem on that crime scene investigation in your quiz. So that's it for this lecture. Sorry it might have been a little longer than normal, but I wanted to get that PCR in there as well. Thanks a bunch.